Now we talk about today, inshallah, we'll solve some problem about first Mendel Law, second Mendel Law, lack of dominance, blood group, abnormal chromosome like Turner syndrome, Clevenger, Down syndrome. To know well how can you answer in question, inshallah. The first problem, when you cross between two P plant, T capital by T small, you can take T capital number 1, T small, encircle the gametes and make fertilization or self-pollination to get first generation T capital T small the resulting ratio 100% at the hybrid impure or tall stem. طب how can you get the second generation? You can another cross between T capital T small by T capital T small to get the second generation and the T capital T small, T capital T small, and you can get T small pure recessive, and T capital T small, T capital T small, T capital T small, you can get 3 T capital and 2 and 1 T small. T capital dominant pure, T capital T small 50, hybrid T small recessive pure. The final ratio 3:1 phenotype and 1:2:1 genotype. 25 T capital at the pure dominant and 25 at the T small pure recessive and 50 T capital T small at the hybrid. This is the first problem. Can talk about the second problem. When you cross between R capital by R small. When you cross between two P plant at the red flower and the white flower, how can you get first generation and second generation? Or how can you determine the phenotype and the genotype? Number one, you can make genetic analysis between R capital by R small as the parent, R capital encircle R small encircle, and after that you can take between R capital R small, you can get to first generation R capital R small at the ratio 100% red but not pure, it's hybrid. First generation 100% not pure, has the hybrid. How can you get to second generation? You can make self-pollination between R capital R small by R capital R small to get the second generation, and separate in the second generation R capital at the gamete, R small at the gamete, R capital at the gamete, R small at the gamete to get R small by R small, a pure recessive, and R capital R small, hybrid, R capital R small, hybrid, R capital, pure dominant. But what about the final ratio? 3:1 at the phenotype, R capital, R small, but R small, pure recessive. But what about genotype? 1 R capital, 25%, 2 R capital R small, at the hybrid, 1 25 pure recessive white flower. Second Mendel Law. We can solve problems about second Mendel Law, but you must listen well to understand what the difference between first and second Mendel Law. When you crossed or answer the problem about second Mendel Law, you can talk about two pair of character like T capital, R capital by T small, R small. How can you read these symbols? This is a flower, tall stem and red flower, and this is plant, short stem and white flower. How can you get first generation and second generation? First generation, we can consider the T capital, R capital by T small, R small, all of them are the parent. And you can get a gametes T capital, R capital, circle T small, R small, to circle at the gametes, and you can make self-pollination between T capital, R capital, T small, R small, You can get T capital R capital, T small, R small. T capital, R capital, T small, R small. What about the ratio 100 hybrid and pure? T capital, T small, R capital, R small. T capital, T small, R capital, R small. What the hybrid? Hybrid, what can you call a tall stem and red flower, but hybrid, not pure? طب how can, this is the first generation, how can you get the second generation? You can make a broad or a square. You can get the final ratio, which you find that 933 to 1. 933 to 1, this is the ratio for second generation for the second Mendel Law. Remember, first Mendel Law talk about 3 to 1, the first generation, or 1 to 2 to 1, the second generation, or second Mendel Law talk about 9, 3, 3 to 1. Don't forget. How can you differentiate between first Mendel Law and second Mendel Law? First Mendel Law and second Mendel Law, all of them are considered the complete dominance, but there are different. First generation in the first Mendel Law, 100 hybrid. First generation in second Mendel Law, 100 hybrid. But the difference in the second generation in the first Mendel Law, 3 to 1 or 1 to 2 to 1. 3 to 1 as the phenotype, 1 to 2 to 1 as the genotype. But what about the second generation in the second Mendel Law? 933 to 1. 933 to 1. All of them are considered complete dominance. What is the difference between complete dominance and incomplete dominance? There are some plants are interhinum plant like a complete dominance when you cross between a red flower by white flower. R capital, R capital, W capital, W capital. What about R capital as the gamete, W capital as the gamete, the first generation, R capital, W capital, but there are difference here in the first generation here, it gives a pink flower or new character appear in the incomplete dominance. Why? Because there is no any dominate to each other. There is no any dominate to each other. There is a opposite than the first and second generation of Mendel Law. When you make self-pollination between the pink R capital W and R capital W, you can get R capital as the gamete, R capital, W capital as the gamete, and you will get W capital white appear, R capital, W capital pink, R capital, W capital pink, R capital pure, R capital appear by 25, W capital 25 as the white, and the R capital, W capital as the pink 50%. The ratio here in the final 1 to 1, but there is a difference between here and there in first and second Mendel. Where is the difference? In the lack of dominance, like interhinum plant, R capital, W capital as the new character appear. But in first and second Mendel Law, there is no new character appear. It may be red, it may be white, it may be tall, it may be short, it may be wide-eyed of eye or narrow-eyed of eye. There is no new character appear. When we talk about blood group, what about blood group? Blood group, as we know, blood group A blood group, B blood group, AB blood group, O blood group. When you cross other problem between A blood group by B blood group. On another question, when you make marriage between man and woman, man has a blood group A, woman blood group B. What about the generation of spelling about this marriage? When you take the symbol A0 or AO by B0 or BO, you can make the steps like first, second, or incomplete dominance. Why? You can take A in a circle, O or 0 in a circle, another circle, P or O as the gametes, and you take O with O to get O, A with O to get AO, BO, AB. The first generation of this problem, you can get all the different blood group. AB 25%, B 25%, A 25%, O 25%. This is an important question when I ask you, how can you get married between two persons to get the all different types of blood group? You can answer to me, when you cross between or you're married between the person, another person, A group and another wife or another person, the female B group. This is very important as a blood group. Another problem on the blood group, when you make marriage between a person carries AB group by A group, you can get first generation. You can make another step as the first, second, A, O, P, A, AP, AA. You can get the first generation about this marriage. Blood group A 50, blood group AB 25, blood group B 25, blood group A 50, blood group AB 25, blood group B 25. This is another problem. And what about 6 linked and 6 inheritance? 6 linked and the 6 inheritance, all of them are called interaction genes. What it means interaction genes? The genes, as you know, it contain a nucleus or nucleus. It contains a sequence of nucleotide. You know, before nucleotide as the chemical structure, it contains phosphate group, sugar like deoxyribose or ribose as a DNA or RNA, and nitrogenous bases. It may be adenine, thymine, cytosine, guanine. We can consider genes which contain a sequence of nucleotide as the chemical substance or the chemical material. All of them can react to each other to appear the character. When you call the interaction genes, we can understand after this word interaction genes, it means the genes can react to each other to appear the character. Here we can solve a problem about a gene interaction as a 6 linked. What it means 6 linked? 6 linked, it means the gene character carries by the chromosome X. Gene character carries by the chromosome X. You know, X as the female, X, Y as the male. When we talk about how can the genetic character carry on the 6 character, we can talk when you make any problem about XY as the male by XX as the female. How can you solve this problem? You can write first the symbol XY as the male and XX as the female. But what about the disease, or what about the genetic character which the 6 chromosome carries it? 6 character like hemophilia disease or blindness disease. Hemophilia, it starts with the letters H. You can write H capital, H capital. This is a normal state, normal state. H small, this is a sick state or sick person. H capital, H small, this is not hyper, but we can carry the disease or carry the disease. I will repeat, H capital as a normal person, H small as a sick person, but H capital, H small as a carrier. When you make marriage between two persons or husband and wife, the husband normal, you can write XY H capital on the chromosome X only, not on the chromosome Y. Why? Because chromosome Y cannot carry any genes. But what about or the importance of chromosome Y? It determines the sex born only. When I ask you in the exam why cannot carry genes, we can say or answer why it not carries any gene, but it determines the sex born. We can get the first generation about this problem when you married between a normal person with or normal husband and sick wife. Sick wife, you can write X H small, X H small. All of them are the parent. What about the second step? You can separate between X H capital in circle and Y in another circle, and X H small in the circle and X H small in the circle. What about all of them are gametes? What about the first generation? You can take one gamete from male X H capital Y by one gamete of female which X H small about. You can take X Y X H small. This is a male infected by the disease, or it is a sick person. What about another X H capital X H small? This is a female, but it carrier. Why it carrier? Because it contain H capital as a normal and H small as a sick, but it can call carrier. Number 3, X H small Y. The third person, X H small Y, infected person by disease or sick person. What about final X X H capital H small? What about the final revision about male? Male, all of them infected by disease, 50 infected or sick. What about female? Female, 50% sick, not sick, carrier. Why carrier? Because the female only carrier because it contains 2 X chromosome. One of them carries H capital as the normal and another carries H small as the sick. But we can call about the symbol X H capital X H small it carrier, not pure or not normal or not sick, it carrier. So what about second generation? When you make married between wife XX carrier H capital H small by a person is the sick X Y X H small. What about? You can take the steps or make the steps before. XH capital circle, XH small another circle, male XH small in circle, Y in another circle. You can get first generation about this marriage. X H small, Y the sick, X H small, X capital normal, XX small female sick, XXH capital H small carrier. What about the final revision between male and female? Male 25% normal, 25% sick. What about female? 25% sick, 25% carrier. All of them are considered sex-linked. What it means sex-linked? It means the chromosome X carries the genes of disease. Don't forget when you write the hemophilia, remember the hemophilia can take the letters H capital as the normal, H small as the sick person, H capital H small as the carrier. Don't forget. What about another solve problems about the sex linked as the color blindness. Color blindness, what it means color blindness? The person cannot distinguish between the colors red and green. So what about the genetic symbols? Symbols C capital, it means a normal. C small, it means a sick. C capital, C small, it means a carrier. What can or how can you get the first generation about when you crossed or married between two persons, wife and husband? Man, XY, sick, it carries C small. By woman, XX carries normal, C capital. What about? You can get at the previous step X small in a circle, Y in another circle, X capital, the circle X capital, another circle. What about how can you get the first generation? You can get male gamete and female gamete by husband and wife. XY C capital, this is a male normal. Another XX capital small carrier. XY C capital normal. Male 50 normal, but female 50. Male 50 normal, but female 50 carrier. When you marry between male sick and female normal, you can get the ratio 50 male normal and 50 male carrier. We can go around the sex influenced. What it means sex influenced? Sex influenced, it is the genetic case which you talk about how can the effect of hormone or sex hormone and the genetic character appear or not appear, and it may be infected by dangerous disease like baldness. What about baldness? When you two person like baldness, it starts by B capital or P capital plus B capital plus B capital plus. What it means B capital? It means in male and female are normal, but B capital plus B capital, it means in male sick, bald, it means female normal. What about B capital plus B capital plus? It means male sick, bald, female it infected by filling hair, not female, not infected by baldness. Why? Because it infected by filling hair only. What do you know about plus? When you write the genetic symbols, B capital, you write B capital plus B capital. What it mean plus? Plus, it indicates the effect of hormone or sex hormone which causes the disease of baldness on male or female. When the sex or secretion of hormone may be more increased, it causes a more infected by male, but it filling hair by female. When you solve the problem, when you cross between P capital plus B capital by B capital plus B capital. In the first case, you can say P capital plus B capital by B capital plus B capital. All of them are similar, but there is a difference in meaning. B capital plus B capital as male, it infected by baldness or baldness, but B capital plus B capital, it means the female normal. Why? Because it contains plus only, not infected by more plus. It may be filling hair, but not now, but after that. When you make any solve a problem about this problem, you can take P capital plus in a circle and B capital in another circle and B capital plus in a circle and B capital in another circle. Let's go with me to get the first generation. Take gamete from male and gamete another from female. B capital plus B capital plus B capital plus B capital plus. What about? We can get the final revision, the final result, B capital plus B capital plus. This is 25. It means male infected and female infected also, but filling hair only, not bald. Number 2, 50% B capital plus B capital. It means in male infected by baldness and in female normal, but 25% B capital, all of them male and female are normal, not infected. This is a more important between sex-linked and sex-influenced. Don't forget when you get answer the question or answer the genetic problem. Now, the final, we can get another problem about the how can you differentiate between Klinefelter syndrome or Turner syndrome or Down syndrome. How can you know all of them? Klinefelter syndrome, Turner syndrome, Down syndrome. Klinefelter, it means 46 chromosome. Turner, it means 45 chromosome. Down, it means 45 chromosome, but it change in somatic chromosome. We can explain now. 47 chromosome, it means Klinefelter. It means 44 plus XXY. 44 plus XXY. But 45 chromosome, it means 44 plus X0. But 45, it means 45 plus XY and 45 plus XX. What's the difference between Klinefelter, Turner? Klinefelter and Turner, the change in the 6 chromosome. 6 chromosome, it means Klinefelter, it increased by 1 chromosome X. X chromosome extra in Clevenger syndrome. But Turner, this is a female case, it charged or decreased one chromosome X. But Down or Mongolian or Mongolism state are the genetic. The change in the somatic chromosome, not 44 but 45 plus XY and 45 plus XX. All of them are different between 3 abnormal chromosomes in the human body. Any chromosome increase or decrease in the human body, it may be led to genetic diseases. Allah subhanahu wa ta'ala create constant number of chromosomes in the human body, 46 chromosome. 46 chromosome, it means that all the individual or person on earth, it contain 46 chromosome in each cell. This is mean that no change between one place and another place, but the abnormal number of chromosome, if the chromosome during the cell division increase one or less one, this is a big problem for all humans body or person. It may be infected by Clevenger like 44 XXY. It may be inverted by Turner female case 44 plus X0. It may be infected by Mongolism 45 plus XY or 45 plus XX. I will give you another advice for your exam. You must read well the question before answer. This is a number one. Number two, you can think well all the words inside the question before answer, because there are similarities or differences between the meaning, between the words, between the shapes of some drawing. Take care. Take care. Read well and think well, and you can answer well. Okay. We will meet about in another time, inshallah. Okay. Goodbye. Assalamu alaikum wa rahmatullahi wa barakatuh.